The single greatest scientific achievement of the last 50 years has improved the understanding of our own species beyond all expectations. As a result, our knowledge of the human race has undergone a major revolution.
Genetics is a very fast-moving subject. When I think about what we knew 30 years ago, to me the 1970s feels like the Middle Ages. That means I also know that what we know now compared to what we will know 30 years in the future is a tiny fraction of that knowledge.
Each of us has a unique blueprint which determines who we are. This blueprint, which comprises 20,000 genes, has now been fully mapped. This knowledge represents a new age of understanding, not only of how we live, but also, in some cases, how we will die.
10 years ago, we had very little information about the cause of these diseases. Today, we know what's causing them. It's a fantastic place to be in genetics, because we now realistically can tackle these diseases using gene therapy. It's a fantastic start point to a very exciting era in genetics as a whole.
The unravelling of the human genome has revolutionised the fight against some 3,000 inherited diseases. Irish investigators are now making key contributions to what is the biggest human science story in the world.
Most diseases are driven by disease genes. We can now switch those off, and that means that we can design therapies to treat many different diseases, many cancers, many skin disorders, eye disorders, neurodegenerative diseases. There are huge possibilities with gene-based medicines.
Doing science is like being on a voyage of discovery. We choose a route and think, this is going to be promising. We go down there, and maybe it'll lead to something very exciting and very novel. Maybe it won't. So we wander around, and we hope we will stumble across something interesting. It's like we're explorers, but we don't have a map. We're making the map. For me, the most amazing thing is that my body contains genes. Those genes have been working continually for the last 3 billion years. I've inherited them from my parents, who got them from their parents, and the genes have been working non-stop for billions of years. They've changed over time, but they're still there. I really want to understand, what are these genes, what are they doing, where have they come from, and maybe where will they be in the future?
Ken Wolfe of Trinity College Dublin is one of the foremost geneticists in the world, and has made the study of the evolution of the genomes of all species his research speciality.
Evolution involves making comparisons over very long periods of time, millions or even billions of years ago. So you can start out with an organism that existed and split into 2 different species, and those species independently became more and more complex on their own branches of the evolutionary tree. And now what I can do in my lab is compare the genes of human and something as different as yeast to it. We can compare those genomes and trace those genes back to the genes that existed in the ancestor of those species 1 billion years ago.
Every single cell in our body contains a copy of the individual blueprint which makes us who we are. This blueprint is written in DNA, and it is unique to each living thing. DNA is actually a long sequence of different arrangements of just 4 letters: A, T, C, and G. The complete sequence is known as the genome, and it is 3 billion letters long. This long line of letters is divided into 23 pairs of chromosomes. Within each chromosome, smaller groups of letters specify every function of our body. These smaller groups are known as genes. Our genes determine everything about us, from eye colour, height, and all physical characteristics, to the likelihood of developing certain diseases. Ken Wolfe is particularly interested in identifying the common genes that humans share with other species.
This is a comparison between a human gene and a fish gene. That's the same gene in two species that are separated by about 400 million years of evolution. So what you see here in the top line, this is the DNA sequence, the A, Cs, Gs and Ts that make up the human gene. And in the lower line, we've got the A, Cs, Gs and Ts that make up the same gene in the fish. And in between them, the vertical bars show bases that are identical between the two. So the fish gene and the human gene, overall, more than 80% of the letters that make up those genes are identical between those two species. And this is because they are both descendants of an extinct species that lived about 400 million years ago. One question that emerges from comparing genomes is where new genes, extra genes, come from. And it had long been known that individual genes could sometimes duplicate, so one copy turns into two, making the genome more complex. In 1997, we started looking at the yeast genome, which was the first complex genome to have had its entire structure sequenced. When we looked at that, we realized that it was full of duplicated genes that were not just made by individual duplications, but that the entire genome had duplicated in one enormous event.
The complete duplication of the entire genetic sequence of yeast proved that at a single point in history, evolution had made a gigantic leap forward. Ken now understood the process by which the genome had become more complex and sophisticated.
I like to think of a genome as being like an old-fashioned factory. There are thousands of workers, those are the individual genes, and they've got specialized jobs that each of them does. Now, the people who own the factory, all they care about is increasing the production, the output from the factory. They're in competition with other factories, and if they can't boost up the output, the factory's out of business. So now imagine that in order to increase the production of the factory, the owners decide that they're going to merge with another factory. So suddenly now you've got 2 workers for every job. So it's survival of the fittest. There's going to be mass redundancy right across the factory. But there is a chance for some genes. If they can find something new and useful to do, they're going to be kept in the genome. They're going to be kept in the factory. So that's how organisms become more complex. That's how the genome, the organism becomes more sophisticated, by gaining extra copies of genes or of workers that find useful new functions to perform.
Wolfe's discovery of the complete duplication of the yeast genome represented a seismic shift in our knowledge of genetics and pointed towards a new pathway in the study of evolution. He and his team had proved the existence of an entirely new mechanism for increasing the complexity of organisms. This work earned them a publication in one of the world's most prestigious scientific journals.
Ken is one of the world's leading analysts on the genome evolution. In particular, he looks at a whole variety of genomes, from little things that crawl around in petri dishes through to big people like us. And he has made his career, really, in showing that quite strange events that have happened maybe millions or billions of years ago, you can see their evidence in the species of today.
The identification of the double helix structure of DNA in the 1950s kick-started modern genetics. In 1990, scientists across the world began to collaborate on the next monumental task: to identify and place in sequence each and every one of the 20,000 human genes. The objective? To map the complete blueprint of a human being for the first time. It was known as the Human Genome Project, and Ewan Birney was head of the European section.
The Human Genome Project was a real international collaboration of scientists worldwide. We had this incredible sequence, all these letters, but what did it mean? And what we did as a group of scientists was really put together the best team we could find in different areas. An outstanding aspect, an outstanding question was there a whole genome duplication at the base of vertebrates when we were ancestors with the fish? And Ken was the person who was the leader in the field who went to investigate that.
In late 2000, as the human genome was nearing completion, we were invited to join the Human Genome Project to ask whether the same evolutionary process, duplication of the whole genome, had happened during the evolution of the human genome. And when we looked, we found that there was not just one, but two successive events of complete duplication of the whole genome right back in the early stages of vertebrate evolution about 500 million years ago. And this confirmed an idea that had been discussed in the scientific literature in the 1970s, but nobody had been able to test it previously.
For his part in the project, Ken had proven for the first time ever the occurrence of two momentous events in the development of the human race. He had shown that at a specific point in time, these events created a fork in the road of human evolution. And one of those roads led to the development of a backbone and a new class of life form, vertebrates.
So the significance of Ken's work is really twofold. Firstly, is that just the fact that these whole genome duplication events occurred. There was a lot of debate, and really Ken has shown that these events really occur in nature. And then from that foundation, one can now speculate, perhaps, about what it means for the organisms who duplicate the genomes. Now, in vertebrates, perhaps this is one of the explanations that there's such a big diversity of vertebrates, from things that fly around, through to reptiles, through to mammals. And so perhaps the fact that all of those share that common duplication is why vertebrates are so diverse.
The fact that we share so many genes with other species, such as mice or fish or fruit flies or bacteria, means that in many, many cases we can use our knowledge of the functions of those genes in these other species and import that knowledge to figure out what the human genes are using. And that's evolution's gift to medicine, this huge resource of information about these other species.
Thank you, please. We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind.
After over 10 years and the work of thousands of scientists, the Human Genome Project succeeded in unravelling the code of 3 billion letters and placing in sequence each of the 20,000 human genes. It was known that human genes were related to functions in the body. Investigators are using this new map to establish what role each specific gene plays, and in turn, how damaged genes are linked to disease. One gene, known to be involved in eyesight, is known as the rhodopsin gene. People who inherit mutations in this gene can develop serious visual disorders. Brian O'Kinneide is one of those people.
For a long time I had difficulties with my eyesight in my youth, and it was in my teenage years when I was out more at night, it became apparent that I couldn't see as well as my friends could see. There would have been difficulty seeing objects around me or seeing people at a distance from me. Eventually, when I was about 17, I went to see the ophthalmologist and I discussed the symptoms I had with them. They sent me for further tests, and eventually I came back with a diagnosis that I had retinitis pigmentosa. In the world as a whole, at least 200 million people are afflicted by visual handicap, and at least 40 million people of those are defined as blind. And many of the diseases which bring about blindness have a genetic basis to them.
Peter Humphreys is a geneticist and a world leader in genetic research into diseases of the eye, including retinitis pigmentosa, or RP. The gene linked to RP is known as the rhodopsin gene, and its protein is located in the pigment of the retina at the back of the eye. The gene was first identified in Peter's research unit in Trinity College Dublin in 1989.
Rhodopsin is a very interesting protein. It absorbs light, it can be sensitized by tiny amounts of light, and it gives us our night vision capability. So if you've no rhodopsin, you can't see at night. Also, other changes in the gene, other mutations in the gene, bring about the devastating disease which causes daytime vision loss as well, and that's retinitis pigmentosa. Major impacts on my life now would be that I find it increasingly difficult to be mobile. You would use a cane if I was walking around at night.
I haven't been able to drive either. I've never been able to drive. And within the last 5 years, my ability to read would have disappeared as well. Even at the moment, like he'll bring me a book and they want me to read the book to him, which I can't do. And you kind of think in the future, well, when he comes home from school and he wants help with his homework and reading something out, I wouldn't be able to help him because I won't be able to see the books that he's using. I'd love to be able to think that in the future, like be able to fully take part in activities with him, like sports and stuff like that. You know, we'll have to see how things are, like how the condition progresses over the next few years and whether that's going to be possible or not.
Jane Farrar in the Trinity Group has been studying the effects of Brian's disease for over 10 years.
So if we look at these pictures here, we've got two retinas. On the right, there's a really dark pigment in the retina, which is indicative that the photoreceptor cells are dying. And on the left, there's a much healthier retina, and there's lovely, healthy blood vessels. These lines here represent blood vessels that are feeding the retina. In this diseased retina, we have dying photoreceptor cells. The blood vessels have attenuated or narrowed, and unfortunately, this patient has probably lost a lot of their photoreceptor cells and actually a lot of their vision.
As the disease progresses, the amount of black pigment will increase as the patient loses more and more of their photoreceptor cells, and eventually they will be completely blind. One of the options for people like Brian is gene therapy.
Gene therapy is important because we're dealing with diseases which until now have been unamenable to therapy. Because we didn't have the sequence of the human genome, we didn't know what was causing these diseases, we had no means of tackling them. Now that we know the cause, we know the mutations which are bringing about these degenerations, we can use that knowledge, we can exploit it in terms of therapies based on that knowledge, and that's what gene therapy is.
Gene therapy means being able to correct the mutations or errors in our genetic code to prevent disease from developing. While it undoubtedly offers enormous hope, it can also raise certain ethical questions.
One of the first concerns that exists in relation to gene therapy is the distinction between therapeutic and non-therapeutic gene therapy. Currently, the only ethically acceptable type of gene therapy is for therapeutic purposes, in other words, for alleviating or for curing disease. However, this, of course, opens up the door for non-therapeutic gene therapy. If I can, for example, change the colour of my eyes by using gene therapy, why should that not be allowable or permissible, seeing that I can achieve the same effect by contact lenses? This, of course, opens the door to other potential uses of non-therapeutic gene therapy in terms of changing traits in the human genome, intelligence, colour of skin, all sorts of other things. And the question that one must ask is whether or not society should allow these radical changes. If we were to abuse the current technology, even if you were to say that perhaps in 20 years' time, if it were to become ethically acceptable, we are potentially looking at the introduction of traits that currently don't exist in the human race. And that opens up the question of what exactly we can do to the species itself.
But what we need to realise is that the diseases are caused by variants of genes, and the genes themselves have been around in the human genome for millions, even billions of years. And they wouldn't have survived in the genome if they weren't doing something useful for the human species. So it would be crazy to think it's a bad gene, let's knock it out and get rid of it. That's misguided. The gene is doing something useful. What we need to understand is what is the normal function of the gene. And then when we understand what the normal function is, then we can think about therapeutics based on that function.
We inherit our genetic makeup, including susceptibility to certain diseases, from our parents. These can be handed down in two forms. In recessive disease, you inherit two bad copies of a gene, and so it simply doesn't work. In dominant disease, instead of two genes which don't work, you have one good and one bad gene, and the bad one dominates its counterpart. Gene therapy for recessive disorders is easier to treat, as the two non-functioning genes can be entirely replaced. In the dominant situation, it's more difficult, as you have to silence one bad gene while leaving the good one intact. What places Peter's work on the cutting edge is that he and his team are using their extensive knowledge to tackle both recessive and the more difficult dominant disease, which accounts for 50% of cases.
But in both cases, we use the same vehicle. We use a little vehicle called an AAV virus. These are harmless little viruses, do no harm to anybody so far as we can tell so far. And they are very, very effective agents for delivery of genetic material, not only to the retina, but to many other tissues as well.
Peter's gene therapy involves adding the correct form of the genetic code into an AAV virus. This virus is then injected into the eye, where it acts as a carrier of the corrected information. It moves directly to the malfunctioning gene and feeds in the new genetic code. The photoreceptor cells in the retina begin to work correctly, and the deterioration is stopped in its tracks. Having proved successful in the test tube, Peter and Jane wanted to see if this technology would also work in animals.
So we're treating blind mice. And we got molecules, or RNAi molecules, that would target the gene, the disease gene in retinitis pigmentosa, and switch it off. This is a picture of that. We have an untreated eye and a treated eye. And really all I want you to look at is the width of the layer of photoreceptor cells in the back of the eye. And the width is much greater in the treated eye because we're retaining photoreceptors. It's much narrower in the untreated eye because the mice are still losing their photoreceptors. It's totally incredible to start to see results like this because the team have been working in this area for 20 years and eventually showing for the very first time that we can have a therapeutic effect. So it's an amazing feeling because you go from literally from A, and we're not quite at Z yet, but we're at the point where we believe we have a methodology which will allow us to treat these animals and hopefully then transfer it later into human clinical trial.
To translate a therapy of this sort into humans, it has to be validated in an animal model. We have to show it's safe and we have to show it's effective. And these are very encouraging times because the therapies we've talked about do seem to be working to slow down the retinopathies in small animals. The job is now to translate this systematically into human subjects. That's the challenge, and that's what we're up to.
Good boy.
Well, what I would like to think is that in the next 15 to 20 years that there would be a treatment, or even better, a cure for the condition, and I would have my eyesight restored back to 20/20 vision, which would be ideal, obviously. But even if they could stop the condition in its tracks so the degeneration was paused, that would be an achievement in itself.
Gene therapy and the use of genetic information offers unimagined hope to the millions suffering from inherited diseases. As a result of the sequencing of the human genome, mankind is on the fast track to a new world of potential treatments for genetic diseases.
The pace of progress has been extraordinary. In 1986, I read a magazine article saying they're going to sequence the human genome, and I thought it was hilarious because I didn't think it was scientifically possible. 15 years later, I was involved in that project. And now, 6 years later still, I get an email telling me I can sequence my own genome for $1,000. It's just phenomenal.
Although there are ethical and practical issues to be resolved along the way, there appears to be a momentum behind gene therapy which is relentlessly gathering pace and carrying us forward towards a new era of medical science.
The diseases we've talked about are not amenable to conventional therapeutics. They don't respond to antibiotics, etc., etc. We can now replace genes and we can now suppress the so-called dominant mutations we talked about. So this is a very exciting way forward, not just indeed for eye disease, but for many other conditions which are of seriously debilitating nature.